We're going to look at two excitable tissues. The first is nervous tissue and the other is muscle tissue. And what we mean by excitable is that these cells can respond to a stimulus and then produce some action. For example, nervous tissue, or the main cells of nervous tissue's response will be to send an electrical impulse to other body parts or other cells in the body, a form of communication. And for muscle tissue, which we're going to see in a bit, they're excitable and they respond to a stimulus, and then their response will be to shorten or contract, causing movement. So examining nervous tissue in general is going to have two major cell populations. The first is the neuroglia, which are several different cell types, and they help support and insulate the actual functional cells of the nervous system. That gives the nervous system its main function, which is to communicate or send electrical impulses that communicate with other cells. They're called neurons. They are excitable and they're conductive because they can conduct the electrical signals. The general structure of a neuron, which we're going to see on the next slide here, include dendrites, the cell body, the neurosoma, and the axon. So you should be familiar with those basic parts. And we'll see them on the next slide here. So here is the actual, in the middle here, this picture right here shows a neuron. And we have the main part of the neuron, the cell body right here. The dendrites are these smaller extensions here that typically will receive information or signals or a stimulus and then generate a impulse, nervous impulse or electrical impulse that always goes out from the cell body towards the end of the axon, where we have your axon terminals over here. So this is a basic description here of the neuron. It's going to talk about the cell body and the dendrites and the axon. It also mentions the cells that help contribute to supporting and protecting the neurons. They're called the neuroglia. And the function of the neuron is to transmit electrical signals from sensory receptors and to effectors. And effectors include muscles and glands that release hormones. And the supporting cells are going to help support and protect the neurons. The location includes the brain, spinal cord, and nerves. And over here you can see the micrograph here. Shows several neurons. And you can see the main cell body of the neuron. So it's pointing at one right there. There's another one here and another. And then you see the processes of the neuron, the dendrites and the axons. And then out here, or throughout where all of these neurons occur, you see these the supporting cells of the neuroglia that are there. I pulled this image off the web, and it just shows the basic structure there with a slightly more detail. Here's your nucleus, the dendrites, the cell body, and then you have your axon, which is long extension there, and the axon terminals that will join up with other cells later. And here you actually see one of the supporting cells called Schwann cells. The Schwann cells help produce this myelin sheath, which is sort of an insulation for the neuron. And then I have another micrograph here showing another example of what nervous tissue might look like stained differently. You can see the neurosoma, the cell body there. You can see the extensions, dendrites, axons, and then you can see the neuroglia throughout the entire tissue sample. Now we're going to move on to muscles. As I said a while ago, they're also excitable, and they can respond to a stimulus, and they are specialized to shorten or contract. There's three kinds. There's skeletal muscle, which is striated or has a striped banding pattern, and they're voluntary, which means you can control it. Then we have the cardiac muscle, which is found in the heart. It is striated. However, we are not under conscious control. We don't have to tell our heart to beat all the time. And then we have smooth muscle, which does not appear striated, and it is involuntary, and we find that in locations like, say, your intestines, and they help move food along. So let's take a look at skeletal muscle. And skeletal muscle is made of tissue. Skeletal muscle tissue is made of these cells that are capable of contracting. We call them muscle fibers. And their description of a muscle fiber is going to be long, cylindrical, multinucleate. So during development, several cells end up fusing together, forming one long cell. And these cells are going to have several nuclei. And then it's going to have obvious striations or striping pattern. Now the nuclei are going to be pushed off to the side because on the inside of the cell is going to be a whole lot of fibers that are involved, protein fibers that are involved in the contraction of the cell. So the function here is it's voluntary movement, locomotion, and manipulation of the environment. They also help with facial expressions and any form of voluntary control. The location is going to be skeletal muscles attached to the bones or occasionally the skin, like in the face, which allows us to make facial expressions. So if we look at the micrograph here, showing the muscle fiber, so there's several muscle fibers here. However, it looks very two-dimensional because it's a thin section or long section through a muscle fiber, which is supposed to represent the diagram on the left, or the diagram on the left is supposed to represent this. You can see the nuclei are off to the sides there. You can see the banding pattern all throughout here and throughout the cell there. So the obvious striations are there as well. So I pulled these images off of the web as well. And so if we look at where like a muscle in the arm, like your bicep muscle in the arm, and the bicep is going to be made of many groupings of these muscle fibers. And if we pull one of those muscle fibers out, this is what it is that we're looking at. That's one muscle fiber. And then it goes into more detail at the microscopic level. We'll study that more later on when we study how the physiology of how muscle contracts. The image here on the bottom right looks similar to the one that's in your lab manual. You see several muscle fibers there. But I wanted to bring this one up here on the top here, because this shows a collection of fibers that show some of them sectioned longitudinally, so you see the long muscle fiber like we've been seeing in the previous images. What happens when you do a cross section through them? This is what it looks like right here. And what you can see is one of these that I'm going to circle within that circle I just drew there. That's one muscle fiber. This is another one. And so again, instead of cutting long ways through, we cut as a cross section, and that's what they look like. And then every now and then you can see some nuclei off to the sides there. So these are your muscle fibers. This image shows you right here when it points to the arrow, and this is from right here to right here. That's one muscle fiber. So now looking at cardiac muscle. Cardiac muscle is different in that the tissue itself of the muscle of the heart is going to be branching, so the cells are not going to be as long. They are still elongate, but they're not as long as individual muscle fibers of the skeletal muscle. They are striated. They're going to be generally uninucleate, which means one nucleus, and the cells are going to interdigitate at specialized junctions called intercalated discs. You can see the intercalated discs. They're these dark, rather dark stripes that you see, or dark bands, darker than the rest of the striations. So all of these right here that I'm pointing to represent intercalated discs. Make sure you know how to spell that. Then you can see a nucleus here and there. So everywhere you see a intercalated disc, that's going to be a separation between individual cardiac muscle cells. So what is the function? As it contracts, the cardiac muscle propels blood into circulation, and it's under involuntary control. And the location is the walls of the heart. So you can see that in the image of the heart here, in our model of the heart. So I brought some images from the web to show that different slides may be stained differently, but you still see these characteristics. You can see the intercalated discs that I'm circling right here in both images, even though they're stained differently. It does appear branched, as you can see some branching going on right here. And so these cells do show branching. Single nucleus between the intercalated discs there. And so they're uninucleate. So it does show the characteristics of cardiac muscle tissue. And then finally we have a smooth muscle. And smooth muscle, here the cells are still elongate, but they're not as long as skeletal muscle. They're going to be spindle-shaped cells with a central nucleus. And essentially these cells are going to be tapered at the ends. Sometimes I've seen books describe them as being fusiform, like fish-shaped. The way fishes are shaped as they swim through the water, it allows them to move through the water more easily. So I like fusiform as a description there. They do not have striations, and their cells are arranged closely to form sheets. The function here is to propel substances like food and urine, or a baby along the internal passageways, and it's under involuntary control. So that's kind of funny, to propel a baby with the uterus, which contracts and helps to push the baby out during childbirth, when during labor, the labor contractions, that smooth muscle that's working to help push the baby out. So the location is going to be mostly in the walls of hollow organs, and this is a micrograph showing the smooth muscle, and you can see the nucleus here. One thing I want to caution you here is be careful that you don't mistakenly confuse this for stratified squamous epithelium, because it does superficially look like stratified squamous. But speaking of superficial, your smooth muscle is always going to be under layers of other or within layers of other tissues, whereas the epithelial tissue, remember, is always at the surface. It's always lining surfaces. So that may be one way to tell apart. If you ever see tissue that looks like this, and then you see other tissue bordering this tissue on the slide, then you're likely looking at smooth muscle. So I found a couple other images to show. Here we have a small intestine in cross section, and lining the inside is going to be epithelial tissue, which we saw earlier. Might be like simple columnar epithelium for absorption within there. But we're going to have some smooth muscle, and the smooth muscle generally is going to have its fibers oriented in different directions. You're going to have the smooth muscle arranged in a circular pattern around to help constrict. And then we're going to have longitudinally arranged layer, which will help shorten the sections of the intestine. And the combined action of both of those layers is what helps move food through the small intestines, called peristaltic action. And so if we were to take a cross section through there, through the intestine, and then put it on a slide, you're going to see what the cells look like in cross section. They're going to look like this, kind of the way the skeletal muscles look different in cross section, and then in long section they will look like this. And that's what we see here. You can see the inner circular smooth muscle, and so the cells are sectioned at long section longitudinally, and then you have the smooth muscle that we cross section through for the smooth muscle that's laying in the long direction there. So the cells are cross section there. I know it says longitudinal here, but that has to do with the direction of the muscle cells themselves. They are laying along the intestine, going the long way, so they're referred to as longitudinal. So these are the circular. However, when we section through it to make the slide prep, the cells are basically in cross section.